Hey, how are you?
Doing good. How are you?
I'm good. What's new?
Yeah, so I was wondering if perhaps we can kind of continue to go over my full leap
Mm-hmm.
exam.
Yep.
Do you see my screen?
Mm-hmm.
Okay. So I think this was the next one. Um,
Mm-hmm.
okay, so yeah, I guess I could just kind of talk through my thinking.
Yep.
So this one, substrate solution was prepared, and then I noticed in the passage that I think was relevant maybe was that there's this 10 grams per liter of glucose, and then they get the molar mass.
Mm-hmm.
Uh, so I'm thinking that because we want to dilute it, I think to the 2 milligrams per milliliter, that like we have to dilute the 10 to this 2.
Mm-hmm.
Uh, and then I'm noticing that this is also in milligrams and milliliters.
Mm-hmm.
But yeah, I'm wondering if perhaps because both of these are kind of like downgraded, I guess, to the same amount, like that it doesn't matter. So,
Mm-hmm.
um, so we need to kind of, I guess, decrease it by like 5 from like 10 divided by 2 equals to 5. Um,
Okay. So I guess what you did was right. I think what they're asking here is that in the cuvette, they put 2 milliliters in it,
mhm.
right? Because you probably did the 10 grams per liter and then convert that into however many um milligrams would be like however many milligrams that would be and however many milliliters that would be, and then you would get your answer. And well, okay, yeah. So tell me more about what you did.
Yeah, I guess I was kind of a little stuck from there because I wasn't sure, like, yeah, like, mm, because they're, I guess, diluted in various ratios of 2 milliliters. Um, oh, but then we're being asked like how much of the original stock.
Mm-hmm.
Um, hmm.
Yep. So you would get your 2 milligrams per liter or per milliliter value. Was that something you were able to get?
Um, I guess I'm like a little bit stuck, like
Mm-hmm.
not entirely sure how to go about this.
Okay. Let's see. So substrate solution of 2 milligrams per milliliter was prepared. How much of the stock solution, which is the 10 grams per liter, was dissolved in a 2 milliliter cuvette? Okay. So let's see, 200 microliters would be, well, 200 times 10 to the negative 6 liters and 1 e negative 3, whoops, liters would be 1 milliliter. So this will be 200 times 10 to the negative, and this will be negative 6 minus negative 3, which will then just be negative 6 plus 3. So negative 3 milliliters, which I guess is just 0.2. milliliters. Okay, let's see. So the way that you got your so was this like a one that you guessed, or did you do like some math to get the 200 milliliter, I'm sorry, microliter value?
I think I might have just guessed.
Oh, okay, okay.
Mm.
All right, let's see then. So the stock solution is the 10 grams per liter. So we can turn that into milligram. Well, it'll be a really, really high amount. So let's see. They diluted it at various ratios in a 2 milliliter working solution before they did these measurements. Okay. Mm, okay, maybe we can do like, let's see, 2 milligrams over milliliters.
Oh, is the way that perhaps the way we're supposed to solve this was like this we're given the substrate solution and then the total volume, and then we can kind of then reverse calculate to see how much milligrams is in that cuvette. So
Mm-hmm.
that's like 4, right?
Yeah, yeah.
Yeah. And
Because
then
yeah, that's why I thought like this value was what you got for this, which would be true for a milliliter, but since they're asking for like how much of the stock solution was in a 2
oh,
milliliter cuvette,
yeah.
you would double it.
Okay, I see. So then because it's 4 milligrams, and then we have to see, given that it's 10 grams per liters, times how much volume will give us to 4
Mm-hmm.
milligrams.
Mm-hmm.
Okay, okay, that makes sense. And then we didn't really have to even pay attention to these units. It's more just like getting the numerical value of 4.
I mean, in this case, since all the units are the same, um, I mean, we still want to be able to pay attention to units and everything like that. But I guess since the units are the same, then, you don't have to as much. But the earlier calculation that I did, um, should be something that you could do. Yeah, that you would be able to do.
Okay, got it. Um, yeah.
Mhm.
Mm, so this
Seems
one,
like a similar thing where the answer was half. So maybe okay, let's see. So based on the figure three, approximately how many moles of electrons are present in a 1 liter solution that creates a 0.5 milli AU, which is, I don't think they mean like gold here. What do they mean here by AU?
probably referring to this
Oh,
signal here.
what do they say? Oh, the sensor response. They probably talked about what it is somewhere else in the passage. The AU.
Um, I think, yeah, it's probably just relevant here, like, and then to do 0.5, and then seeing that's like roughly 0.75 of substrate concentration.
Okay. All right, maybe we don't need to know. Okay, let's see. So approximately how many moles of electrons are present in a 1 liter solution? Okay, so 0.5. Oops. Okay, that's like okay, so I guess what we can see what we can see here is that our value is larger than this zero point 0.5,
Mm,
and well, I guess here in the middle would be like 7.5,
mhm.
right? So our ant wait, because then that would then rule this out. So okay, let's see. Substrate concentration. Oh, okay, okay. What? So substrate concentration, 0.5, response. And then they tell us that, or they're asking us how many electrons are present in a 1 liter solution that makes this sensor response. What, okay, let me look at like the pat, like I want to see like what's actually happening here. So, because my thoughts are that this might be like, have something to do with the number of electrons that could be like transferred in like a redox reaction. So they mentioned 2 electrons here. So that's what I was thinking.
Mm.
Because typically we don't deal with, like, you're not going to really deal with moles of electrons, um, unless you're going to be dealing with redox, in which, because typically whenever you have your metals that turn into cations, like sodium plus and all the other ones in that period that have a plus charge, which means that they are going to lose 1 electron, and then you can have the ones that are like, calcium, magnesium, and stuff. So usually for metals and redox and things like that, you're going to have like a 1 mole or 2 mole amount of electrons. So this seems like what they're telling us here is that in this redox like reaction, 2 electrons are present. And so whatever you did, so was this one where you guessed also, or was this one where you got some idea?
Perhaps what I think I would do here is like after calculating the 0.75,
Hmm.
um, so that gives us substrate concentration, and then we have to convert that to moles. So then I would have divided the 0.75 milligrams.
Hmm.
Oh, I guess I would have to convert that first to grams if that's necessary. So
Yeah,
I
that
would
seems like,
multiply it. Yeah.
uh-huh, go on.
Yeah, I would have multiplied it then by like 1,000 liters or wait, 1,000 milliliters because that's how much is in 1 liter.
Okay.
Which and then um divide that by oh wait, because then I'd still have to divide that by 1,000 milligrams to give 1 gram. So the 1,000 doesn't really matter. So it's still 0.75 grams.
Yeah, because, well, tell me again about the number you just said.
Like, I guess I was trying at first to convert the 0.75 milligrams over milliliters to just grams over
Hmm.
liters. But then I realized that it's just the same, right? Like,
Okay. Let me see the part that gave that redox reaction again.
mm,
Okay, so um polyaniline, okay, oxidation states. All right. Okay, they made a biosensing platform using polyaniline with an immobilized enzyme. It's based on reaction one, in which a reducing agent converts the imine group into an amine by donating 2 protons and 2 electrons. So we have this reaction happening here. Now, so this reaction seems like it's separate from the reaction that an enzyme and a substrate would normally have, right? Because they're
mm,
saying that they took so I guess the polyaniline would be is that the substrate? Let me see. Or was the glucose the substrate? Let me see below, on the left. Let's see.
yeah, so I guess this. isn't it based on the reaction?
Got it. Okay, so what we see in figure three is Michaelis-Menten, like, kinetics with this characteristic logarithmic curve. And what this does is really just tell us like how gives us information about how the enzyme can take the substrate, which I guess they're like literally telling us here, but like that's typically what you're gonna see in the x-axis for Michaelis-Menten curve. So this tells us about the substrate that the enzyme turns into a product, right?
Mhm.
But this is separate from the polyaniline, which they um reduced or was it oxidized? They did a redox reaction with the polyaniline to turn the imine into the amine. So that's separate from like essentially what figure three is telling us. So so figure three, I don't even think is going to really matter as much as well what they told us about the redox reaction that they mentioned earlier in the passage. Yeah. So the reducing agent, so it gets reduced. So basically, this glucose gets or sorry, the polyaniline. Wait, what's this? EB? Okay. Wait.
Oh, so I guess it's the idea that we, from like this, determine like how much glucose is being used, and then each glucose produces two electrons.
Let's see. Okay, fully reduced state. It's called lead, and I guess an intermediate is EB. Okay, reducing agent. The imine. Okay, let me see what they're saying here. Go up a little bit over here. Yeah, like way above. Like even higher. Okay, so let's see. Okay. Yep. Next. Okay, and then they did this. This and the solution is following.
Yeah, so I guess the idea was to find the moles of glucose first, and then each one produces two electrons.
I'm gonna put on my like regular glasses for this.
Mm.
Okay. All right. So this is the way that we should do this. Um, let's go up to the question. Okay. Now, all in all, like when you read these passages, I know you mentioned something about like reading the questions first and then going to the passage. Now, like that might be fine, but remember that like the test is designed to like kind of trick you in that way. So we want to separate first the enzyme, like, enzyme substrate gets turned into a product from the enzyme, and they said glucose was the substrate. And that's what they're telling us in figure three. That figure three is just the Michaelis-Menten curve for an enzyme. Now that's completely separate from anything to do with changing the number of electrons of something. So when you see like approximately how many moles of electrons are present here, I want you to be thinking redox. And that's also how the passage starts. It starts by talking about, well, essentially redox here. And here in the part that you highlighted, we have, and then earlier here they talked about like the reduced state is LEB, and the EB, I guess, is like the intermediate oxidation state, but we could just say like before it got fully reduced, right? And now it doesn't seem to say this explicitly, but I was thinking of it and it turns out to be the case. Now, if EB is the thing that's getting reduced into LEB and nothing else is being like mentioned, the only other thing that's there has to be the reducing agent, right? So so the reducing agent in this case happens to be glucose, and that's the only reason we can connect it to figure 3, which would give us, yeah, that would give us a glucose concentration. Okay? So so overall what we want to see here is um whenever they ask you anything about moles of electrons or number of electrons, it's going to be tied in with a redox reaction because redox reactions involve, transfer of electrons with your half reactions and things like that. And and the redox reaction that they told us about that's earlier in the passage is the one that tells us that it's 2 electrons. But it also tells us, not explicitly, but it also shows us that glucose is the actual reducing agent. So what we essentially then do is we look at figure 3, we get some value for the amount of substrate, which is glucose. And that's also the amount of reducing agent in that, you know, redox reaction. And the redox reaction is gonna, like, the stoichiometry of that redox reaction is seems like it's balanced with like 1 as a coefficient for each. And they said for each, there's going to be a 2 electron reduction. And so that's how we can connect it to um, this value here.
Okay, that makes sense.
Yeah. So
Mhm.
so a lot of times the explanations can make it even more confusing, but just in terms of the thought pattern here, uh and that connection that I made, that's the important part. Like because for instance, if in that redox reaction glucose was not the reducing agent, then figure 3 wouldn't be of like we wouldn't be able to use that to connect it to anything.
Okay.
Yeah.
Okay. And then the Y value in figure 1
Oh, and
changes.
then this one.
Oh, is that the Nernst equation?
Yep.
Okay.
So this little n value, like or I guess symbol, stands for moles of electrons,
Mhm.
right? And like this cell potential is, the sum of like the the reduction potential and the oxidation potential. So it just link those two things in your head. So when you see the words like moles of electrons and stuff, yeah, think about redox. Redox is like it's like going to be everywhere on the test anyway, so. Okay, now this could be related to the fact that so again, it's something that they talk about here,
Mhm.
but here is where, like, when I was explaining the previous problem, I was saying that this EB is an intermediate oxidation state,
Mhm.
right? So so at EB, it's partially um, either oxidized or reduced. So that's then connected to this part.
And I guess I should have also read the fact that it says like when y equals one, it's a fully reduced state, so it's moving more towards a reduced form.
Hmm.
Yeah, I don't know why I chose oxidized, but
Okay. So I guess for that one, then you had the right idea in that you notice that it said like intermediate.
yeah. Okay, this one makes sense. And then this one I was a little confused on because I thought that like fructose. Oh wait, also when this so this shows glucose and fructose, right? And then that oh my
Mm.
gosh, that disaccharide is known as s- wait,
Sucrose, like
sucrose.
table
Sucrose,
sugar.
yeah. Mhm. And then so usually when they bond and make that glycosidic bond, is it that fructose is like flipped over, right?
Okay, let's see. So in general, for sugars, what you need to know
Hmm.
is like you need to know how to draw the straight chain form of glucose. Is that like the Fischer projection
Hmm.
for glucose, right? So like be able to draw the straight chain form and then understand how that straight chain form can turn into that ring through and whenever you see hydroxyl groups, I want you to think nucleophile, and whenever you see carbonyl groups, I want you to think electrophiles.
Hmm.
And you're gonna have the fifth carbon for glucose, at least the fifth carbon's hydroxyl group nucleophilically attack the first carbon's carbonyl group to form that ring structure. And like 0.1% of glucose in our bodies exists as that straight chain. Um, the rest exists as rings. But do you know like the alpha beta anomer?
Yeah, I think the alpha one is where it like points down like this and then beta points up,
Yeah,
right?
so it'll be related to the last, like if this is up and
Hmm.
the other one is up. Um, yeah, so the beta one is going to be 64% of the glucose in our bodies, and like the 36% will be the alpha form. And the reason for that is that in the beta anomer, when you draw it as a chair, all of the hydroxyl groups are in equatorial positions, and that means that they're further, and that makes it more stable because the hydroxyls are not as close to each other because they would repel one another. So that's glucose. And then to know the what's an epimer?
That's where it differs at like one
Yeah,
chiral center. Mhm.
correct. Um so you they expect you to know the C2 epimer,
Mhm.
which is mannose. And you could think like man with a gun, that. And the C4 epimer, which is galactose. Okay. And all of these are aldohexoses. Galactose and glucose, disaccharide of, oh yeah, and then when for your, so every single functional group, or yeah, you're always gonna have to know like Okay, so for instance, what's this function? It's a little weird one, but
Is that like considered a hemiacetal?
yeah, very good. And then if this was an R, that would just be a
An acetal? Or
or
wait, um,
what's like the other possibility, like an acetal would be similar to like an aldehyde.
oh, is it like the ketone?
Yep.
Is that what
Yep.
it's called? Okay.
So it'll be a hemiketal,
Uh,
okay? And uh hydroxyl groups, unless they're tertiary, like secondary and primary, can be oxidized into carbonyl groups. So if something can be oxidized, it can reduce other things. So there's a like either there's like Tolin's test, there's Benedict's test, and
mhm.
these test for reducing sugars, which something like this, like a your hemiacetal or hemiketal, something like that will test positive as a reducing sugar. Now, when you have like, another one with whatever, it's four, like this is one position, this is one, two, three, four. So like a 1,4 linkage. When this and this is going to form through dehydration synthesis, but this then becomes, well, just something like this. So now instead of it be so if it was a hemiacetal before, now it's a full-fledged acetal. And if it was a hemiketal before, it's now a full-fledged ketal. And now acetal or a ketal no longer has a hydroxyl group, which means that it can no longer be a reducing sugar, and therefore it will test like negative, for the Tollens test or the Benedict's test. And from all of that, we can see that for the most part, monosaccharides will be reducing sugars. Polysaccharides will be, will not be reducing sugars, except like you could have a really long chain, but the one that's, let's say like this is a long chain, but this one on the right is the end of the chain. Well, that end will indeed have that hydroxyl group. So if you have like glycogen or whatever, like cellulose or any like polysaccharide, you
Mhm.
could still test positive just on that last uh monomer. Okay? Okay, so like galactose, glucose, these are all, oh yeah, and then fructose is also going to be, do you think it'll be a hexose or a pentose?
So it'll be a pentose.
All right, now that's a kind of tricky, like a tricky kind of question that I asked you because it looks like a pentagon.
Oh, oh, but it still has the six carbon. It's just like forming a five carbon ring.
Yeah. So now why would it form a five, membered ring? And it's because, okay, so like this would, sorry, that looks bad. Like, whoops. So for an aldohexose, the first carbon's going to have an aldehyde up here, and that's what gets nucleophilically attacked. But if you have a ketohexose, then the carbonyl group, by definition, cannot be the first carbon because a ketone would need, carbons on both sides. So that's the only reason why, and it's still the same kind of thing where the fifth hydroxyl group nucleophilically attacks the carbonyl group, but this time it's just that because it's a ketone, or well, the first carbon cannot possibly be a ketone, so the second carbon is a ketone. That's why it takes on this five-membered ring uh form.
And then for the linear structure of glucose, I'm like struggling to remember it. Like, is there like an
Yep.
easy way to?
So I say it's just if you do the middle finger. So I'll show you what I mean here. So and you imagine like this is your hand.
Mhm. Oh, okay.
Hmm. And then so if you had a C2 epimer, this OH, the on the second carbon would go here, and then
Oh,
that's the man with the gun.
gone. Okay, makes sense. Okay. So in this one, I think I was a bit confused because I thought like just looking at this, like I think this is like wait, yeah, this would be the like considered the anomeric carbon, I guess. But it seemed like it was pointing down, so I thought that the fructose would also be alpha.
So I just want to ask you,
Mm.
tell me what happens when this forms a ring.
So like the fifth carbon would attack the aldehyde either from as the top or bottom. That's what creates either alpha or beta.
So which carbon or
So
which hydroxyl?
fifth one, like or like this one down here.
Hmm.
Mm.
Yeah. So it's going to do that because you know throughout nature and stuff a six-membered ring is really stable. But um okay, so for instance, this oxygen, which
Mhm.
one is it?
So it would be the one that attacked, right?
Yep.
Uh-huh.
So this one, right?
Yeah.
And then this, I'll use orange. So this one, so yeah, it's gonna attack, and it's not really like a thing that matters necessarily like from which side because there's really only one side, it's really just going to attack that, and that is like a double bond, so it's not like you know, that its movement isn't gonna determine whatever anomer you get. Um, but you're gonna get some of each anomer, and what they can do is it's different from resonance. Like resonance is like if you drew like resonance structures, you would do this type of thing with the arrowheads on both sides, but a single line. And that's because in resonance, it doesn't go back and forth. It exists as both. But for the different anomers, it's something called I think it's a tautomerization, or it was it mutarotation. It was one of those. But it actually does go back and forth between the two. Um and then the beta is preferred for those reasons that I said before. And um yep. What were we asking?
Yeah, I was like wondering why this fructose is considered beta because I thought that if this O is kind of pointing down, then um it should be alpha.
Okay, let's see. Um, okay, so we can get rid of these, and then there's both beta, both, and one alpha and one beta.
Like, yeah, like I thought it would be alpha fructose here, but yeah, I just wasn't sure why it's considered beta.
Let's see, running up, carbon two. Wait. Seven, oxygen group on carbon two pointing up. Okay, so I think this might be related to the fact that so the anomeric carbon here is the first carbon, right?
Mhm.
And oh, this is interesting. Okay. So um yeah, so we count this like that, but for fructose, if you think about it, you would actually start because since you okay, so since the numbering has to go through the longest path,
Mhm.
that's going to include like it's not going to start from something that's just on one side of this oxygen here, right? Like the way that it
Uh,
starts, yeah. So like,
it would start like with this carbon technically.
yep, so this,
Oh, but
that
would
would
we
be
still, yeah, but would we still consider carbon 2 as the anomeric or would it then be? Okay, so I guess like carbon 2 is the anomeric carbon for fructose. Okay,
yeah, yeah.
that makes sense. Um, yeah, and then I was also wondering if maybe we can like kind of switch to, I was doing this flashlight like right before this and I was honestly just really like stuck with how to break it down. So can I like kind of go over like my thought process and like maybe give like any recommendation to tweak it? Because yeah, I get like kind of I guess lost with like these type of passages, but um
NFTs, that's funny.
yeah, so reading this, I was I guess trying to make some notes on like so it seems like they're talking about NFTs and then um that the translation can either be done by this cap at the end of this region or it can do it without the cap through IRES that's also located in here. And then I continue to read, and then I see that um
Okay. All right, here, let's go up to the
mm,
top. So, yeah, so this is good to go over. So,
mm,
um, and this is typically, I'm guessing this is like a BB passage, right?
yeah.
Yeah. So these passages have will always have like a trillion acronyms, and what we can do is we can just write proportionalities for the acronyms, just so we can see like if this goes up, this goes up, this goes up, this goes down. Um, and and then also recognize those buzzwords. So I'm just gonna read it real quick. So Alzheimer's, um, NFTs. So Alzheimer's AD proportional to NFT, right? So if you have more of one, you have more of the other thing. So then they say NFTs, aggregates of hyperphosphorylated tau proteins that have dissociated from microtubules. So this could be proportional to, I'm just gonna write like, I usually write like a phosphate as like a P with a circle around it, and then maybe like tau here. Um, and then like if I wanted to do an inverse proportion, it would be 1 over, so I'll say like um micro, just to mean, or like micro T, to mean that these NFTs are composed of these like hyperphosphorylated tau things that are no longer attached to microtubules, right?
Mm, okay.
Okay, and then they say neuronal degradations, AD symptoms, like ironically, like the clinical stuff is like what they don't test as much. So we want to just be able to break everything down into like the basic sciences. Okay. In general, ribosomes initiate translation of mRNA by binding the 7 blah 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 cap at the end, at the 5 end of this of the untranslated region of the mRNA molecule. Uh, so we also want to know, just in like it's like background information, that um when DNA gets transcribed into mRNA, um well, in addition to RNA being inherently less stable because it has that extra hydroxyl group, and we talked about hydroxyl groups acting as nucleophiles. Um, and because mRNA is going to leave the nucleus, the DNA will never leave the nucleus. Um, because outside of the nucleus, there's a lot of exonucleases, which can start to break apart um, nucleic acids from the ends, hence the exo. So you're gonna have like a 5 prime cap uh, on the 5 prime end of the RNA to protect that end, and then a poly A tail on the 3 prime end to protect that. Now they're being more specific here by giving it a name like the 7-methylguanosine cap, blah blah blah. But we just want to like assimilate that with what we already know. Okay. However, researchers believe that the tau protein translation and initiation can occur without recognition of this cap. Okay. So what I'm thinking here is there could be some unchecked uh translation um of an mRNA molecule uh because like it can happen even though there isn't the cap that you would normally uh need. I mean, like this isn't something that we know, but you see how it says like in general, and then
Mhm,
they say researchers believe. So
mhm,
this is likely going to be some type of hypothesis that they're going to create an experiment for at some point, and you're going to see that type of um, order, like a clinical thing, just to give it some relevance to like the MCAT, and then the science stuff, and then some type of phenomenon that researchers are trying to like learn about, and then they'll create an experiment, and then you'll see the results and stuff. Okay, so they believe that the protein translation can happen without that cap. Okay, this alternative mechanism, referred to as cap-independent translation, indicates that ribosomes are recruited by IRES, internal ribosome entry sites, generally located in the 5' UTR of the mRNA sequence. Okay, now another thing is that they're not necessarily saying that if you do not have the cap, that you can get you could still get translated. That's why they're saying without recognition, you could still have that cap. Um, but it ribosomes can translate the protein or translate that mRNA without needing to recognize that cap,
Mhm.
right? And that's why it makes sense that this says internal ribosome entry sites, because that could just mean that that it could just attach the mRNA like, well, I guess what they're telling us in the UTR region instead of the cap region. Okay. All right.
Mhm.
Um, oh, and then like I would say like, all of this stuff would likely be proportional to IRES. So all of these things, we can relate to one another instead of like keeping it in our heads because it's like impossible to keep that type of information in our heads. Okay, to determine the initiation mechanism of tau mRNA translation, neuroblastoma cells of AD mice were transfected with two different DNA constructs, figure one. Construct one contained the 5 UTR of tau mRNA extracted from cells displaying NFTs. So these are, it could be like perhaps like a positive control. It's like this is the mRNA um, for something that we know has this bad thing that we're looking for. Okay, construct two included the 5 UTR of the beta globulin gene, which normally codes for a plasma protein. Okay, so now this is just going to be something, it's not going to be like a negative control because we don't know for a fact that it's not going to work, but Um, but I would say construct one is going to be like our positive control because we know that that's going to, you know, be bad, right?
Yeah, I guess like when I was first reading this, I thought that construct two was going to be like some sort of control because I thought like plasma protein is like not what we're focusing on. So like maybe they just want to like make sure that whatever like um the translation could happen.
Yep. So that could be a way to establish a baseline, but it depends on what you're trying to measure if you think about it, right? Like if you're thinking about a hypothesis for these NFTs, right? Um, which are so yeah, so if you're trying to think of like a hypothesis for NFTs in this case, then it would make sense to use an NFT as a control, right? And then as a positive control, something that creates NFTs.
Hmm.
So it ultimately depends on that. If the hypothesis was, trying to find out something about uh something that was normal, I guess, then you can because even though we just use the term kind of like liberally by saying a control, and that usually means like a baseline, we just want to think of uh a little bit deeper into is this because every control can be a positive or like will give us more information if it's a positive control or a negative control, right? So if this construct 2 was something that we knew does not cause NFTs, right? If
Hmm.
that's something we knew, then that can be a negative control,
Hmm.
right? But from what I see so far, um, I mean, it says normally codes for a plasma protein, so maybe it could be similar to that. Um, but yeah, I would always look for like that directionality when it comes to controls.
Okay. Yeah. And I guess like how I then kept reading was So then there's like two luciferase reporter genes. So I'm thinking like this is not related to like what was discussed before. So they're again testing maybe something else even.
So when
Um,
you see this figure, I also wanted to ask, like, when you see the figures and stuff, like, do you like skip them or do you like what do you?
I mean, I look at it, but I don't really like take the time, I guess, to like fully dissect it yet, unless like the passage asks about it, and then I'll kind of go back and then see if I need to interpret it more.
Yeah, so I would say give yourself like maybe like uh I would say maybe like look at it
Mm,
because if you think about it, like the purpose of figures and things like this is to make it easier for us to understand, right? So I would say to try to look at it to understand what it's trying to show you. And maybe that should take like 5 or 10 seconds. If it takes longer than that, then move on and come back to it if you have to. But for something like this, where we have just essentially two of the same things with like one thing that's different, that can also make it very easy. So we see whatever promoter, blah, blah, blah. We see exactly what we were kind of expecting a little bit earlier when I said the poly A tail. Um,
mhm.
Now, the only difference here seems to be the UTR region, right?
Yeah, and I guess like from what I can gather from this image, perhaps like I'm thinking that like the Ranella, which is one of the things that they were mentioning, the luciferase reporter is occurs before the untranslated. So if they were to test like IRE as like I guess this wouldn't be affected in perhaps like if the IRS or IRES is working or not, like this would be translated versus not because I don't know, like if it starts here, then it'll continue going forward. Like I guess that's kind of like something that I'm thinking from this image.
Got it, got it. Okay, okay. So yeah, that IRES does change things in terms of how I was looking at it before. So it seems like both of these are are able to be recruited by the internal ribosomes,
Mm.
right? So okay, that changes things. Good to know. Um, okay. Okay, okay.
Yeah. And then I guess if I were to keep going down, so then I read um so Lucifer student expression from constructs is monitored in presence of a synthetic. So they added a cap. Translation depended on ribosomal binding to the cap, but okay, so then they're distinguishing between these two that this the Ranella one depended on the cap being there, which makes sense because it's before. But the one that comes after required the IRES ribosome, which also makes sense. And then looking at this graph, um, I see like as we increase the cap, um, it shows that the Ranella signal in and then it distinguishes between the two constructs. So
Yeah.
const one.
Okay, let me see this. Okay. Luciferase gene expression from the construct was monitored in the presence of various concentrations of the synthetic cap analog. Figure 2, translation of this depended on ribosomal binding to the cap. Okay, so the luciferase cap. So this can tell us that, and then I guess if it's gonna bind to the internal site, it's not gonna bind to the cap. So this would be inversely proportional to luciferase, right? And then let's see. But translation of photinous luciferase. Oh wait, there's two different types. Required. Okay, hold on. I gotta change this then. Okay, so Ranella. Okay, so the Ranella one happened. when it binds to the cap. And if it binds to the cap, it's not an IRES, so I put an inverse proportion there. But the photonus one happens when it's in the IRES. So then I could say this is proportional to P luciferase, and then I could say this is inversely proportional to P luciferase. Um, so this is just a way for us to keep track of all of this.
Mhm.
Okay, let's see. And then figure two, so we have the construct one square, construct two triangle, and we have Phytonus and Ranella. Okay, so let's see if what we wrote out in the proportionality lines up. So we see that construct one is the one that has the tau one, which we should expect to have the Phytonus luciferase activity, but not the Ranella one, right?
Um,
Based on this over here.
so oh, because yeah, the tau is linked to RES. I guess also like a struggle that I have with these type of passages, like I forget all like the relations between them, and then I have to like circulate back to like the first paragraph to remind
Yep.
myself.
So that's why when we read the passage, we write this stuff out. So you
Hmm.
see how I wrote like the IRES thing
Hmm.
is inversely like proportional to the Ranella luciferase, but proportional to the Photonus luciferase, right? So, and then construct one, I mean, I could just say construct one is proportional to all of this. Construct two is proportional to all of this. So, construct one, we should see more of the Photonus, which we see over here with the straight line, right? But we should see less of the Ranella, which we see here going down, right?
Hmm.
Okay. And then construct two, we should see the opposite. So construct two, we should see um less of the Photonus and more of the Ranella, right?
Yeah.
And you see how like I wrote this stuff before like looking at the figure, which means that we can like figure this stuff out like logically like this, and then the figure could just help us verify that. So let's see. So construct two is triangle, yeah, it's higher for the Ranella, which lines up exactly with this.
Hmm.
All right.
Yeah. So I guess like the conclusion here is that like so construct one with tau, um so it and then that's okay, construct one is proportional to the P one, the phonetics, so it requires RES. So construct one basically needs IRES, whereas construct two,
Oh yeah, I forgot about that part. Okay, so the Ranella one involves the cap. Okay. That makes sense. That's why the Ranella one is less happening in construct one, right?
yeah.
And the Photonus um does not require binding to the cap,
Mm,
right? So we see less of the Photonus in the one with the cap, right?
yeah.
Okay, so for this question here, ribosomes interact with, so they should react with the uh Ranella one cap. Ranella one was what construct two. Yeah. 5 cap of construct two or the IRES of construct within constructs 1 and 2.
Mm,
Let's see. Data in figure 2. Oh wait, so all the data is like basically 0 for construct 2 with Photonus, and Photonus is the one that does not use the cap, right? So then it should be, so for the, okay. Okay, so Photonus does not need the cap. Photonus happens more in construct 1. So construct 1 should work in the IRES, right?
Um, yeah.
Okay.
Yeah, Photonus needs the areas.
Yep. So that would be,
I can eliminate B probably.
oh,
Oh,
well, I think construct 1 was the one that, wait, sorry. Construct 1 was
oh wait,
the thing
construct
that
one has
didn't need the
high,
cap.
but I guess I'm getting confused because it seems like with a lot more of the cap, there's more luciferase activity. So construct one does need the cap.
Let's see, construct 1, Photonus, broadstone by it. Okay. Photonus, construct 1. So IRES for construct 1, and it seems like construct 1 also had some activity for, wait, Ranella for the cap, whereas for construct 2, it's mostly the Ranella, which means the cap. So construct 2, Ranella cap. Yeah. Unless
So
they said something different later on. But
yeah, so I guess like construct one is able to both have the cap and the IR. Yes, so probably D then.
yep,
Uh,
because we could have the cap for both of the constructs. Yep.
okay. Um,
So yeah, the real lesson here is the proportionalities.
mhm,
Like like there's no real alternatives. Either you try to remember it all, but working memory doesn't let us have this much information in our heads at the same time, or you have to continuously go back to the passage. And like sometimes students say things like, like how am I going to have time to write this out? And the answer is, there's no alternative. Like either it's going to take more time if you do the other stuff. So like the way that I read that, as I was writing it, I just kept writing these proportionalities out so that I don't need to know really what NFT means, right? But I know if it goes up, then, we're gonna have more of the Ranella, right?
I
So that's essentially how we answer that.
I see. Yeah.
Mhm.
Oh, and I guess, yeah, you have another class, right? So,
Oh yeah, let me see. We could do like we could do like 5 more minutes.
okay. Um, yeah, I guess. Um, I'll try to like interpret figure 3. So in the second study, researchers use siRNA. On the top of my head, I honestly don't really know what siRNA is, but I'm just going to try to continue. So target initiation factor, a peptide translated in cap-dependent manner. So here, maybe I would put a proportion between this and cap. That facilitates the binding, and then proportion sign again to this. So yeah, like I would say ELF4E proportion to ribosome plus like ribosome maybe draw an arrow to M70PP cap. And then it seems like they're yeah, assessing this in siRNA treated cells. Um, and then so from the figure, I'm seeing that. So the negative control, it seems like yeah, the experiment is working because there's signal. Um, and then it seems like there's, I guess, less of EF2K when ELF4E is there. So maybe this downregulates that, but it seems like this upregulates tau.
Okay,
So,
so do you see I put the L43 or 4E thing here?
uh-huh,
Because they said that it was a peptide translated in the cap-dependent manner.
uh-huh.
And like this is where the cap stuff is,
Hmm.
right? And um okay, now siRNA is silencing, like it's basically so if they used siRNA to target this ELF4E, that ELF4E is no longer gonna work,
Oh, okay.
right? And if we didn't know that, I think that's something you could have also figured out from the like how the negative control is like that. I mean, I don't know why the siRNAs like for
Hmm.
both, but okay, let me see. Expression of this factor, elongation factor. Oh, okay. So this is the same as that. Okay. Elongation factor is this. So this is the initiate handles the initiation factor. And then this is the elongation factor. And then tau, okay, in the siRNA. Okay, so if the ELF4E contributes to binding to the cap, and you got essentially uh rid of that, you should not have initiation happen, right?
Hmm.
So this thin band here makes sense. Um, for the negative control, it's a little confusing because it says siRNA, like for both, but in siRNA tube cells, okay. But okay. Let's see. So in the elongation one, there's even less of the initiation. Or no, sorry. Okay, I think the SI, this is a weird graph or chart or whatever, but I'm trying to think like, okay, does negative control, like, okay, so they're all treated with the siRNA.
So siRNA
Yeah.
would knock out kind of ELF4E. So without ELF4E, I guess EF2K is dependent on ELF4E because it's also not expressed, but then tau seems to like still be expressed without.
Oh, okay, I think I understand. So the siRNA only targets the L4E, the one that's responsible for initiation. So for the one that's involved in elongation, with the siRNA, like, yeah, you're not going to have as much of the ELF, like the initiation thing, and you're going to have, I guess, a smaller amount of the elongation, but you're still going to have some amount, I guess. And then the tau, it seems like it doesn't matter at all, and that would make sense because they said that the tau thing, well, for people who have AD, but these are non-AD cells. So the tau, okay, these are non-AD cells, so the tau is not being like hyperphosphorylated. So I guess that's kind of like the actual control here. Like the elongation or the initiation is not going to be affected um, for the overall tau protein.
Okay,
Okay.
so I guess like I would
Yep.
choose D then perhaps because it seems like it's dependent on ELF4E, which we read in the passage is also cap dependent. So if they're all related together,
Let's see,
then
only binds to the mRNA transcripts of proteins that facilitate this, signals for the degradation, that elongation, that doesn't seem it. Translated, cap independent. And they tell us here that it's cap dependent for that. So that can't happen. Then this is translated via a cap dependent one. And I guess that's why we see a thinner band for when you get rid of the one that causes initiation. Hmm. Dependent. All right, let's see. Okay. See your list. Okay. Negative control. So this is whatever this is
Mhm.
with the uh silencing of the elf 4E. Okay. And makes sense that we have a small amount of this. And then they talked about how this binds to the cap, first step, but if this is absent, cannot be made into a protein. Induced by siRNA leads to noticeably reduced expression of the elongation factor because it's mostly absent, unable to bind to the cap dependent process. Okay. Okay. So yeah, I think this is just very hard to interpret, but I guess if they're saying that, or if they're showing us that there's less of the amount of the elongation one, this, when you like damage or you inhibit the initiation factor, and since the initiation factor is cap dependent, and when you got rid of that initiation factor, this one shows a much thinner band. I think that could help us figure out that the elongation factor one, the 2K one, is dependent on the L4E, which they tell us is cap dependent. So that means we could deduce that it will be cap dependent.
Mhm.
Okay. And so yeah, I think this is hard to get like straight up the way that we just did it. So I would use a combination of essentially that and then process elimination because I noticed that the other choices were easier to kind of eliminate. So for instance, like this, they tell us right here, right, that this is cap dependent, right? So we can get rid of that. And then this, the rest of these, so this says siRNA only binds the mRNA transcripts of proteins that facilitate whatever IRES uh ribosome binding, but we said before, or we knew from before, that IRES means that it does not bind to the cap, right?
Hmm.
And this siRNA binds to the thing that binds to the cap. So this, in other words, is kind of saying what C is saying,
Hmm.
right? And then B says degradation of proteins that facilitate elongation of tau proteins. But we see that whichever, I mean, first of all, siRNA, it's not going to signal for the degradation of a protein. It's going to silence an mRNA. Something that will signal for degradation of proteins is like ubiquitination.
Hmm.
But aside from that, the part that says facilitate elongation, we have identical bands for the tau protein.
Hmm.
Um, whether you are silencing, the ELF4E or not. So yeah, that would rule out B. So I think ultimately understanding this, I mean, I think I think just from seeing like the Jack Weston passages that the figures and stuff are a lot more ambiguous. I don't think it would be like that so much for like the other tests. So like for most tests, like I think looking at the figure can help out a lot. But I think for this one, the figure was on the confusing side. So we want to use like a also like the process of elimination.
Okay. Yeah, that makes a lot of sense.
Hmm.
Okay. Um,
So I would say, um, all in all, to try out like that proportionality thing,
mm-hmm.
as you read those passages with lots and lots of acronyms. And yeah, um, and then I guess like, yeah, making like the connections that we made before for like how a figure is different from what they said before about like oxidation and and then like when if they're talking about moles of electrons, that's going to be related to redox. Yeah, anything that you wanted to